Good morning, everyone. So, next practical that we are going to start with it is bioassay of histamine. So, it is like continuation of the things that we have studied in our theory classes. So, today we will be finishing about the bioassay of histamine, and the next practical will be the bioassay of serotonin. Okay, so let's see the purpose of today's experiment, that at the end of practical class the student shall be able to know about the advantages and disadvantages of matching bioassay. Then second is know the calculation of potency of unknown test sample by using matching bioassay method. The third is the know the pharmacological action of histamine, which we have already covered in theory classes. And lastly, to know about the guinea pig ileum preparation and its uses in bioassay. Okay, so these are the four purposes of today's practical class. Okay, so out of it, the first one, that is matching bioassay, we have already studied about it like a very long back. It was like the first practical that we have covered about bioassay. If you don't remember, it is my suggestion that you should go and revise that particular thing. Okay. So terminology, what is matching bioassay? It is a type of graded bioassay in which the potency of unknown test sample is calculated with respect to standard by matching the responses between test and standard compounds. Very simple. What we will be doing, as the name suggests, we will be matching the response of the test with that of the standard. And what will happen because of that? We will get to know what is the concentration of test sample in that particular given dose. Okay, we already know the concentration of standard, and by matching the test dose height with that of the standard, we get to know the test concentration of that particular test sample, test dose, and from that we can reverse calculate that how much quantity of the drug is there in the test sample. Okay, so it is very easy to determine, but this particular method, it suffers from a certain shortcomings. These shortcomings we will be studying in further slides. Okay, so description: histamine is present in almost all the animal tissues, mostly within mast cell and basophil granules. Then tissues rich in histamine, they are skin, gastric and intestinal mucosa, lungs, liver and placenta. Non-mast cell histamine is present in brain, epidermis, gastric mucosa and growing regions. Histamine is also present in blood, most body secretions, venoms and the pathological fluids, and it is now known to play important physiological roles at various locations which we have just mentioned above. Then histamine, it produces effects by acting on the histamine receptors, that is H1, H2, H3, H4, which are present on the target cells. All of these receptors, they are G protein coupled receptors, and they modulate the CAM, that is cyclic adenosine monophosphate, to effect downstream signaling. Okay, so the main actions in human through H1 receptors are increase vascular permeability, then vasodilation and contraction of most smooth muscle except blood vessels. And through H2 receptors, the actions which we get are the stimulation of gastric acid secretion and the cardiac action. So all the four receptors, they are implicated in the inflammatory response in some capacity. Okay, so this all we have already studied, that's why I went too fast in this particular two slides. Okay, so the concentration of histamine in biological samples, it can be studied by using different bioassay methods. Okay, so depending upon pharmacological action, histamine can be assayed by the first one, that is the contractile effect of isolated ileum of the guinea pig, and second is fall in blood pressure of anesthetized and atropinized cat. Okay, so these are actually two methods of performing bioassay of histamine. The first bioassay method, it deals with the effect of histamine on the gastrointestinal tract. Okay, and the second effect, it is because of its effect onto the cardiovascular system. So what is atropinized cat? Atropinized cat means the cat in which we have already injected the atropine. You know the atropine, what is its pharmacological category, what effects it produces, everything you know. Okay, so we have already administered atropine into the cat, and then we are administering the histamine, and we are checking that the fall in blood pressure. So the fall in blood pressure will be the point of interest in this particular experiment. Okay, and the cat here, it is very much alive, but it is just anesthetized for the purpose of experiment. In the first experiment, we are killing the guinea pig and we are isolating the ileum. So the first experiment will be the in vitro experiment, and the second experiment will be the in vivo experiment. Okay, so objective of today's experiment, to determine the concentration of the histamine by matching bioassay method using guinea pig ileum muscle preparation. Okay, so the principle, guinea pig ileum is very useful preparation for the bioassay methods, and it is more sensitive to histamine. So in most of the histamine related experiments, you will find that we use the guinea pigs. The reason too is that the guinea pigs are very much sensitive to the actions of the histamine. So it becomes very easy for us to observe the effects the histamine produces onto the various tissues. Okay, so contractile response of the histamine to ileum is due to presence of H1 receptors. Matching bioassay is a simple method in which a constant dose of unknown test substance is matched with the varying concentrations of the standard drug. And how we do that? By trial and error method. Okay, so we are keeping the test substance dose at a constant value, and what we are changing is the standard drug concentration values. Okay, so whichever concentration of standard, the height of it matches with that test, that standard we will consider for further calculation. Okay, so it is said that till the exact matching response is observed, but trust me, when we are doing the experiment in actual laboratory condition, there are some minor changes observed in the response height between the test and the standard, and that minor change or minor difference in the response height it is considerable. We won't get an exact match. If we get exact match, then it is better option. But if we don't get an exact match, there is some slight difference between the two responses, then we can consider the same. Okay, so this is actual real life example. So a corresponding concentration of test substance is then calculated. So first thing or first objective is that we have to match the response height of the standard with that of the constant dose of the test. Okay, so once that gets matched, we can calculate the concentration of test substance. Okay, so here response is matched at only one dose, so it does not need DRC of test and standard compounds. Okay, so as we don't need to draw the DRC or to take the DRC of standard and the test, the reason is that we require very small sample volume, and that is the like pro point of this particular method, that if we have very little substance with the very little test substance and standard substance in our hand, then in that case we have to go for this particular experiment, that is matching method. Okay, so requirements. So animal or tissue, animal we require guinea pig whose weight should be between 500 to 600 gram, and the tissue which we have to extract is the guinea pig ileum muscle. Okay, so the drugs which we have to use, that is standard solution of histamine whose molecular weight is 111.15, and the test solution of the histamine, so concentration is unknown over here, and the PSS that we are going to use, it is the thyroid solution. So equipments we have to use, that is chymograph with drum and smoked paper, then student's organ bath, then isotonic frontal writing lever, then aerator with aeration tube, and some other accessories. And some other requirements we require, that is tuberculin syringe, then beaker, stopwatch, petri dish, dissection box, suturing needle, and the thread. Okay, so general requirements are these. So procedure. The procedure we can actually divide into like 2 or 3 parts. In the first part we will be actually talking about that how to kill the animal, how to extract the tissue from the animal, that all sort of things we are going to consider. And in next, there is how we are going to perform the experiment, that we are going to consider. Okay, so in first, that is we have to prepare the stock solution of thyroid, then also we have to prepare standard stock solution of the histamine, and then different concentration of histamine we have to prepare by the serial dilution method. Okay, so we have to sacrifice 24 hours fasted guinea pig by standing on the head, and we have to do the carotid bleeding. So we have to fix the animal on the dissecting board by tying its legs. So we have to open the abdominal cavity by a small horizontal cut followed by a vertical midline incision, and we have to expose the abdominal organs. So we have to locate ileocecal junction by lifting the cecum. Okay, and then we have to go upwards up to 8 centimeter from the junction, and we have to cut the 2 to 3 centimeter long segments of the ileum muscle, and we have to exclude the terminal 5 to 8 centimeter which contain excess of excitatory alpha adrenergic receptor which may interfere in the study. Okay, so in order to locate the actual ileum, you need to have very good knowledge of the anatomy of the abdominal or the gastrointestinal tract. Okay, so you know at the ileocecal junction, from cecum onwards large intestine starts. Cecum, prior to the cecum we have the ileum. Okay, so that's why we have to identify first of all the cecum, and then we have to go upwards, that is 8 centimeters from that junction, and we have to cut 2 to 3 centimeter long segments of the ilium muscles. The first 2 to 3 centimeters only we have to cut, rest we have to exclude. Why? Because it consists of the alpha adrenergic receptors. Okay, so next is immediately we have to put this muscle preparation into the petri dish, which contains aerated warm thyroid solution. Slowly we have to remove the mesentery, that is the white part which is attached to the muscle, and we have to gently clean the lumen of the ileum by passing lukewarm thyroid solution through it with a pipette or a syringe. Simple process. Okay, because that ileum may consist of the fecal material, so we don't want that fecal material to contaminate our tissue or interfere with our readings. That's why we have to clean it from internally. Okay, so we have to mount the preparation in the inner organ bath, which contains the thyroid solution, that is around 20 ml, and we have to maintain it at 37 degrees centigrade. We have to tie the bottom end of the muscle to the hook of aeration tube and the upper end to the isotonic frontal lever by a thread without closing the lumen. Okay, so lumen should be open. So only one end of that particular tissue we have to tie with the thread, and the other end should go to the isotonic frontal lever. So we have to adjust the magnification of response to 7 to 10 folds. We have to aerate the tissue with 2 or that is O2 or carbogen slowly. Okay, so that is 40 to 60 bubbles per minute. So stabilize the tissue for 30 minutes by applying a tension of 0.5 gram weight which is attached to the lever, during which wash the tissue with fresh thyroid solution once in every 3 minutes. So this procedure is like common to all of the experiments that we will be doing by using the student organ bar. Okay, so 5-minute time cycle we have to maintain, contact time of 30 seconds, and that what is the 5 to minute, how this 5-minute time cycle we have to divide. At 0 second we have to remove the weight and we have to start the drum, and we have to record the baseline for the 30 seconds. Then at 30 seconds we have to add the histamine to inner bath, and we have to record the response for 30 seconds, that is the contact time. So at 60 seconds we have to stop the drum, we have to add the weight and wash the inner tube. Okay, so at 180 seconds we have to give the second wash. At 300 seconds we have to give the third wash, and we have to remove the weight and start the drum again, and we have to take the baseline. Okay, so record the responses of the test compound, that is unknown concentration of histamine, with gradually increasing volume till obtaining a response T, which would lie on the linear portion, that is 30 to 70% of the CRC. So fix the response obtained due to volume of T. So we have to record the several responses due to different concentration of standard histamine solution on the tissue by trial and error method until response is of the standard solution it matches with that response obtained due to volume of T. You can see we have taken 3 responses of the test, that is 0.1 ml, 0.2 ml and 0.4 ml, and then gradually we are going to increase the standard dose sample, that is from 0.1 ml to the 0.8 ml, and at one point we will get to know that the 0.8 ml of the standard it matches with the 0.4 ml of the test. Okay, so approximate matching we have to do, not the exact matching, and we have to then subset or divide this dose into two. We have to take half doses of the test and half dose of the standard. That is half dose of test, that is 0.2 ml, and half dose of standard, that is 0.4 ml. The logic behind is that if the 1 unit of test matches with 1 unit of the standard, then half unit of test of the test will match with the half unit of the standard. Same applies with 2 units of test should match with 2 units of standard. We are just confirming whether there is an exact match between the two or not. Okay, so you can see the next dose is 0.2 ml of the test. What was the original T? It was 0.4 ml, and 0.4 ml of the standard. What was the initial volume? That is 0.8 ml. And in next two, what we have done is that we are taking exact double quantities of it. Okay, so 0.8 ml of the test and 1.6 ml of the standard, and ideally both should match. Okay, so for further confirmation we have to record the response due to half and double the volume of the T and S and find out whether they are equal or not. Okay, ideally they should match if the original T matches with the original S. Okay, so this is the calculation. So calculate the concentration of test compound as follows. So this is the what to say formula for it. Concentration of T is equal to volume of S divided by volume of T into concentration of S. We know this concentration of S as we have prepared the standard solution. Okay, so where S is a response of standard histamine solution and T is a response of test histamine solution. And also we have to calculate the potency ratio of the test compound by the formula is potency ratio is equal to concentration of T divided by concentration of S. Okay, so this concentration of T we will get from this earlier formula. Okay, so observation table will look something like this. Histamine solution, we have test solution and the standard solution. We have the concentration of standard and we are supposed to find out the concentration of test. Okay, and the volumes they are mentioned like this: 0.1, 0.2, 0.4, and of the standard whatever volumes we have taken, that we have to mention over here. And also we have to mention measure the response height in millimeters. We don't have to express it in centimeters, we have to express it into millimeters. Okay, and we have to write over here. Okay, so inference. So matching bioassay, it is a simple method in which comparison of potency between test and standard compound is done. The concentration of unknown compound may be calculated by matching the responses which are obtained due to test and standard histamine solution. Then also the potency ratio of the test compound may be calculated. Okay, so what we are getting from this particular method, that we are able to find out the concentration of test sample, as well as we are able to find out the potency ratio of the test compound to the standard. Okay, by very easy method. So these are all questions to you. So please read these questions, write these questions in your notebook. I will wait for 1 minute for you to write down all these questions, and just like last experiment, you have to submit these questions into the Google form which I will be sharing into the Clever now. Okay, so 1 minute to you to write down these particular questions. Okay, I hope that you got all the questions in your notebook. Solve it and submit it as early as possible. I will create Google Sheet at the earliest, and I will put the Google link, that Google form link into the Clever so that you can do the submissions. Okay, so that was all for the bioassay of histamine. We will start with the bioassay of serotonin in few more minutes. Okay, thank you.